Assalamualaikum and warahmatullahi wabarakatuh, and very good afternoon to everyone. Okay, first of all, welcome to Immb International Webinar Series. We're really excited to have all of you today. A big thank you to our speaker, Dr. Manfred Kriechbaum, Senior Research Scientist, Graz University of Technology, Institute of Inorganic Chemistry, Graz, Austria, and all the staff for joining us. Okay, without taking too much time, I will invite our director, Dr. Wang Sukwan, to say a few words. Please give all welcome to Dr. Wang.
It's my honor to introduce Dr. Manfred Kriechbaum. Thank you for coming back again. I believe that this is your second time delivering a talk at IMB at the Institute. All right. So Dr. Manfred is from Austria, and he is specializing in small angle X-ray scattering and structural studies of biological and material systems. He is currently works as a senior research scientist at the Institute of Inorganic Chemistry, Graz University of Technology. He holds a master and a PhD in physical chemistry from Karl Franz University, Graz. His academic career includes research roles at the Austrian Academy of Sciences and a postdoc fellowship at Princeton University in the biophysics groups. All right. Dr. Manfred research primarily focuses on small angle X-ray scattering, including time-resolved experiments, structural studies of proteins in solution, and the development of small angle X-ray scattering. He played a role in the conceptual design of the small angle X-ray scattering beam line at Electra, right? It's Electra in Trieste, and has conducted the experiment at international synchrotron facilities such as the ESRF. Actually, I'm not sure what it stands for. ESRS, DSY, and NSLS. Okay. He also contributed to the development of lab-based SAXS system, including their worldwide installation and personal training. Beyond academia, Dr. Manfred has held industrial positions, including a research stint at Raychem in the United States and a decade-long association with Hackers X-ray Systems R&D department in Graz. His expertise has earned him Mascade Fellowship in 1991 and 1992. He has authored over 100 publications, including recent works on fuel cell catalysts, biomaterials, lipid membranes, and nanostructures.
Okay. Thank you very much for the introduction, and to have the privilege to be here the third time or the second time to give a talk. It's always nice to come here to Malaysia, and I always enjoy here the hospitality and also the cooperation which I can do here with the universities. Thank you. My talk today will be about molecular nanostructures studied by small angle X-ray scattering. This research I have been done already since over 25 years. Currently, I'm at the Graz University of Technology, and we are in operation of a core facility of such instrument which is applying this method to many scientific fields like biomolecular nanostructures, but also material science. But today I will more focus on biomolecular nanostructure. To structure my talk, there will be at the beginning some basic introduction, so you are familiar with this technique, because this is not a straightforward everyday use technique, so it's more a niche technique, but it has a lot of potential application you can do, and you can solve also many problems or structural problems with this technique. And then I will also give you some examples of current research which can be done. but without going too much into the detail. So the main thing is that students and postdocs should get an idea about the technique, and maybe an idea how they can apply it to their problems. Okay, there will be introduction, basic principles, the lab and synchrotron SAXS instruments I will introduce, and then some basics about nanoparticles and dilute systems, shape modeling, and later on I will come to the applications like proteins, liquid crystalline systems, phase transition, high pressure SAXS, time resolved SAXS, and so on. Okay, let's first start and just look how you categorize this technique and for which problems you can apply it. So SAXS is not a spectroscopic scattering, but it's elastic scattering of X-rays, and it's a direct imaging method, but not as microscopy. It's direct imaging in the reciprocal space. So I will explain later what it means, reciprocal space. So opposed to optical microscopy, and it is probing structures in the nanometer range. So this is very important, the range where you can probe investigate your nanostructure. Here you have a comparison of microscopical techniques like transmission electron microscopy and cryo-EM, SEM, and optical microscopy. They covers a range from about 1 nanometer to micrometer or millimeter. SAXS is this range here with this green highlighted. So it's in the range from 1 to 100 nanometer. So what is beyond that size, like in the micron, you have to use other techniques like light scattering, dynamic light scattering, or dimensions lower than 1 nanometer, you have to use diffraction, Xray diffraction methods. Here you can also see the nanostructures which are in that range, like from the single atom, from molecules up to molecular assemblies of proteins, of viruses, of bacteria, and then organism. So just remember our window in size is from about 1 nanometer to 100 nanometer. So you have to be always very aware of the limitation of this method. Just to give you a short example or animation how this works. You have an X-ray source which sends out some X-rays. The X-rays are being collimated, so you have a very fine beam, and this beam hits then the sample, which is a source of scattering. And the scattering is being then detected by a position-sensitive X-ray detector. One thing you also have to remember, the incident and the scattered X-ray beam have the same energy. So it's not the spectroscopy where some energy transfers occurs. It's elastic scattering without any energy transfer. And the scattering is caused by the electrons in the sample or in the molecule. So if you have some sample with a homogeneous electron density, you almost get no scattering. You have a flat baseline. But as long as you have inhomogeneities in your sample, so high and low electron density, this gives then rise to a scattering pattern. The scattering pattern looks like here to the right. You can get the two-dimensional scattering pattern, which are either isotropic or anisotropic. If they are isotropic scattering pattern, then you can make an integration, a radial, and get a one-dimensional scattering pattern, which is intensity versus the scattering angle, which is displayed here. So you just integrate the spectra to get the one-dimensional scattering curve or diffraction pattern, intensity versus scattering angle. Here, an explanation of reciprocal space and the real space. So with light, you can have lenses to magnify your object. So you have your object, you have your lenses, you magnify it, and then you get a magnified virtual larger image. But for X-rays, you don't have lenses. There are no lenses to exist for X-rays. So in that case, by scattering, your picture gets stuck in the middle of the lens, so to say, and this is a reciprocal picture. And the other side of the lens in microscopy transforms it back into the real image. But here you have no lenses, so you have to make a mathematical transformation. This is called Fourier transformation, to transfer your scattering pattern, which is a reciprocal picture, back into real space. This is illustrated here. Your picture scattered picture in the reciprocal space, applying Fourier transformation in mathematical process, you get your real space picture. Good. Maybe just some historical facts, so you know in which time frame this technique developed. So remember, 2 centuries ago, in 1895, the X-rays were detected by Röntgen in Germany, so 1895. And maybe 40 years later, the first experiments or measurements were done with small angle X-ray scattering. And there were mainly 2 groups in the 30s and 40s of the last century. One group was in Graz by Professor Otto Kratky. So he was an expert in theory, instrumentation, and also application. And he lived and worked all the time in Graz. So he was actually still alive when I studied there in Graz. So he was one of the pioneers. And second person was from France. There was a group of France around André Guimier. And he was also a pioneer in that theory. He developed also some instrumentation and contributed in theory. For doing an SAXS experiment, you need also an X-ray source. And there are 2 sources available, either in a laboratory or at a synchrotron. A laboratory source you can buy maybe for $500,000, or you can build a beamline in an accelerator, in an electron or positron accelerator. They can build a beamline, which is much more expensive, of course, but you also have some advantages. So in the lab source, your exposure time is in the minute range. And with a synchrotron, you can make your diffraction pattern already in microseconds, because the intensity is 1,000 times higher than in the lab source. There's also a difference in spatial resolution, in sample environment. And in the lab, you have always access, and at the synchrotron, you maybe get 2 or 4 weeks access, and there you have to finish your experiments. So it's limited access. Where are those? Here is a picture where you have such synchrotrons. In Europe, I'm using mainly the synchrotron at Elettra, which is in northern Italy. And if you look to Malaysia, the closest one is there's one in NUS in Singapore, but they are more in spectroscopy and So nothing's scattering. The closest one would be here in Thailand, the SLRI, or maybe in Taiwan, or maybe also in Melbourne, Australia. So there you have this large-scale facility with a high-intensity X-ray beam. They can make very good SAXS experiments. This is just a scheme of the synchrotron at the electra. So you have here the ring where the electrons are being accelerated in a magnetic field. The diameter is about 80 meters. And there you have about 25 different beamlines. So you can also build beamlines from the wavelength range from infrared to X-rays. You can also have spectroscopical beamlines, scattering beamlines, and also other ones. Using all these different techniques like photoelectron emission, imaging, scattering, absorption, diffraction, lithography. Okay, so let's go back to the principle. The principle of small angle X-ray scattering. The underlying mathematical formula is Brett's law. Brett's law basically says that the scattering angle of your scattered X-ray is inversely proportional to the nanostructural size. The sinus scattering angle equals to your wavelength divided by the nanostructure, by the size of the nanostructure. This means it's inverse proportional, that small particles scatter to large angles, and large particles scatter to small angles. And we are focusing on small angles, or focusing on the large particles. So we have to use small angle X-ray scattering, and not powder X-ray diffraction. With powder X-ray diffraction, this is wide angle. You're studying atomic distances. But we are more interested in nanostructure or nanostructural assembly of molecules. So our range is, I would say, from 1 to 100 nanometer. So it means our angular range is, if we're taking a wavelength of 1 angstrom, is about 0 to 10 degrees. So all our information is between 0 and 10 degrees. This is small angle X-ray scattering range. Which samples you can investigate by SAXS? You can investigate solid, powder, crystalline, amorphous, space-like viscous samples, liquid, even gaseous samples. So this is a big advantage to crystallography. And because in crystallography, you have to crystallize your samples before you study them. Here you can use any aggregation state, and you don't have to do any preparation. You don't have to stain it. You don't have to make it freezing or make them conductive. So it can use as they are. The sample amount is very little, so it can measure 50 milligrams or 1 microliter you can use. The thickness, if you have solid samples, should be around 1 millimeter. And it's a non-destructive method, so after the measurement, you could reuse your sample again for some other experiments. Here is Hedge. What can be measured? There can be measured either isolated nanoparticles in a solution or in a matrix, but there can be also a amorphous or porous system where you have amorphous or porous samples like polymers, coals, ceramics, or wood. Or you can also have crystal or liquid crystalline samples, which we see then later 1 or 2D or 3D crystalline samples. So they are solid dissolved in the aqueous solvent, and you can also name it the soft condensed matter. So we go on. So the difference between crystallography and SAXS. In diffraction or single X-ray diffraction, you have to take crystalline samples, so you have to get a crystal. And in a crystal, all molecules adopt the same configuration or the same position with respect to the X-ray beam. In that case, you don't get a diffuse scattering pattern, you get sharp diffraction points. And these, if you do the gate evaluation, you can get then atomic structure, because you have the information of the phase information and the intensity information. And by that, you can solve the atomic structure of your molecule, if it can be crystallized. In opposition, SAXS, where you have your nanoparticle in solution, they adopt any conformation with respect to the X-ray beam. There's a spatial and the temporal averaging of your nanoparticle. So you're losing this information of the phase information. You only have the intensity information. And this gives you then a diffuse scattering pattern, so no sharp single X-ray spots. And the result, what you can get, is a one-dimensional curve. And from that, you can only get the envelope of your nanoparticle, the envelope shape, due to the loss of information, due to this spatial and temporal averaging of your structure. But on the other hand, you can investigate any sample in the natural environment, and you don't have to crystallize your sample. So which information you can get from your scattering curve? So this again is a scattering curve. Here's an angle 0, and here is a maximum angular range. And this is a scattering curve, so intensity versus scattering angle. You have three regions. In the inner part of your scattering curve, you get information about the size. In the intermediate range, you get information about your shape. And the outer region of the scattering curve, we get information about the surface. So size, shape, and surface. Good. This is now a more complex slide. You start here in the reciprocal space. So you have your scattering pattern. And how do we get into the real space? You have to do an electron transformation, a Fourier transformation, to get your one-dimensional P of R function. So by Fourier transformation of the scattering curve, you get your P of R function. What is a P of R function? The P of R function, you can understand, is a histogram of all possible distances within your particle, within your nanoparticle. This is, for instance, here's a sketch of a conformational change of a protein. Here you would see, this is only a simulation. This scattering curve would change that way, and the P of R function, the Fourier transformation of it, would change that way. And you can immediately recognize when the structure is compact, you have a symmetric curve, and when it's in the elongated state, you have two maxima, which is assigned to these big ends of this protein. Maybe I'll give you a better example later. Okay, reciprocal space, real space by Fourier transformation. You get your one-dimensional P of R function. Here again, in a static picture, for different sizes, for different shapes, the scattering, like if you have a very symmetric particle, you have a very sharp maximum minima, and if you have a very anisotropic nanoparticle, you have a very diffuse scattering. So how does it look like in the real space? The sphere has also a very symmetric BFR function. And the hollow sphere, in the hollow sphere, you only have large distances. So the large distances will have a maximum at the diameter of this hollow sphere. So this is this blue curve. So what I want to say, what this picture is telling you, that in the reciprocal space, you have not a real understanding how your structure looks like. Only in the real space function, you have an idea or have a clue that how the particle might look like. Like what I told you, here you have the long distances have a maximum at the diameter. So this means this is a hollow particle. Or in this two double sphere molecule, you get this pink curve. So you have two maxima. So it consists of two particles of two masses which are a distance apart. So understanding is better in the real space than in reciprocal space. Okay, here's just an animation how this scattering curve changes the space. Large particle scatter small angles and small particles to large angles. But that way, the scattering curve will change. Here, if we have fully dispersed particles, the scattering curve gets more diffuse. Monodispersed, sharp maxima minima, polydispersed, diffuse scattering. This is an example. So you can also study a polydispersity. If you have the same shape, so this was in a publication we did maybe 5 years ago. These were nanoparticles of some indium particles synthesized. And we nicely get some nice size distribution of these nanoparticles by small angular scattering from this SAXS curve. This is how the change of the scattering curve of BFR function would be if you change the shape. If we go from an oblate to a prolate, the curve would change that much, and here the BFR function is the same. This is just an illustration, a simulation, to get a feeling how the resulting reciprocal space and real space would look like if we had this shaped nanoparticles. So the general strategy for solving structural parameters in small angular scattering are that way. You have your experimental data. You evaluate the structural parameters like radius or radius of gyration. You can determine also the mass and the maximum diameter. So size, mass, and diameter. And then you make a model in the real space. You transform this model back into reciprocal space, into your experimental space where you got your scattering curve, and then you compare it with your experimental data. So what does this mean? Here you have your experimental scattering curve, and then you try to find in the real space a model which fits your experimental scattering curve. So the red curve is a corresponding scattering curve, and the green is your experimental. Then in that case, you end up with an elongated particle, because this red curve fits the green curve. You have to search a model in the real space which fits your scattering curve in reciprocal space. So once you have found your model, you compare it with the data which were obtained by another technique like with crystallography or with NMR. So many proteins were crystallized, and the structure was solved with atomic resolution. They deposit it in a data bank or database, and there you can retrieve this data and compare it with your shape, with your low resolution SAXS data. Like in that case, this is lysozyme. So you overlay both results, and then you can say if it fits or if it is plausible with the model or not. So there's another example. Okay, I've got such shape simulation. You start out with some random shape, and then you change your shape till it fits your experimental data. Then you take the real structure from the crystallography and compare it with your low resolution model from scattering. Okay. So now you might ask why I'm doing then even SAXS if I have high resolution data from crystallography. The thing is that if you do crystallography, you have to crystallize your sample or your protein, which is done in a very unphysiological condition. So in high salt concentration, you have to crystalline your protein, which might adopt a similar structure like in a native buffer, in a native solution. So we have done here a comparison of some protein. This is a crystal structure, and in a native buffer, we get a slightly different solution. So there is a discrepancy between solution structure and crystal structure. It is not in every case, but in some case it might be that there's a difference because in a native buffer, it is more likely to adopt its real native conformation. And what you can also do with proteins in solution, you can add some substrate easily and then see the conformational changes if we add some substrate. This you cannot do so easily with crystallography because every time you have to crystalline your samples with the substrate and then see the changes. Good. If you can also denaturate your protein, and then you can also fold the denaturation from the folded state to a partially unfolded state to totally unfolded. So if you do denaturation, you can time resolve, you can follow your denaturation of a native protein in solution. So far, we were only talking about static features, but to understand the function of a protein, you also have to study its dynamic. So this is just a comparison. This is a skier, so this is a world champion skier, but just by studying his body, you have no idea about his technique, how he is skiing. So you also have to study the static fixture or structure, but also how they change when they do some action, like this protein here. Okay, and for that purpose, you need time resolved Xray scattering. So you have to apply some force or some change on your protein and study then the time slice changes. Later, I will give you some examples of that. Yeah, already here's one. So I have to. Okay. Okay, this is not moving. Okay, usually this molecule here should move. What you have seen at the beginning, so here it does not move. At the beginning, you have seen this animation. So then you see the changes in the scattering curve and in the real space. Let's see here. So here we see it better. This is a simulation when of the spike protein of the COVID virus, when it's being docking to the ACE receptor of the cell. Because the COVID virus has these spike proteins, and when it's docking to this ACE receptor of the cell, it can fuse with the cell and integrate into the cell. And here is some simulation how this would happen. So the spike protein then opens the pore into the cell, into the membrane cell of the living organism. And this would cause such changes if you would see it. I mean, this is only a simulation, but if you were able to monitor this action, you could easily record it with small language, with timers of small language scattering. So let's go from protein to lipids or amphiphilic molecules. Amphiphilic molecules, like from chemistry, they consist of the amphiphilic, of the hydrophobic part and the hydrophilic part. So this is why it's called amphiphilic. And depending on the shape of the molecule, of the single molecule, depending on the solvent where you put it in, either oil or water, or also on temperature and pressure, it can adopt different nanostructures. So it assembles it in itself in a nanostructure. So it can have three-dimensional infinite nanostructure, lamellar nanostructure, or even a hexagonal, two-dimensional hexagonal nanostructure, depending on the composition of the lipid, as I said, on the water oil content of the solvent and temperature or pressure. So again, you can in that way produce single shell vesicles or multilamellar vesicles. If you have a single shell vesicle, you will get such scattering curve, and from this scattering curve, you can determine the electron density of this bilayer. So here, of the single shell vesicle, the unilamellar vesicles, you make the Fourier transformation and get the electron density of the bilayer. So in the interior of the bilayer, you have a low electron density, and on the outer surface, which is exposing itself to water, you have a high electron density. So this way, you can also study if we insert some track, if it is in the bilayer or if it is in the inside of this unilamellar vesicle. So where the track is located, you can determine the location of your track. You may also be able to make not only single shell liposomes or vesicles, you can also make multilamellar. So the infinite number of multilayers of these bilayers, always separated via water layer. You can make this one-dimensional lamellar, two-dimensional hexagonal. In that case, the water is in this cylindrical cores, or you can also make three-dimensional complex structures. Here again, this multi-layer lamella. So you have bilayer water. So there's infinite stack. And with SAXS, you get the information about the long-range order. You can also do simultaneously wide-angle scattering. With wide angle, you're drawing the shorter distances, so you get information of the ordering of the lipid, of the lateral ordering of the lipids. You get information via wide angle scattering. So this is a case sample of a hexagonal structure. You get, of course, a different diffraction pattern. From the diffraction pattern, you can easily determine the morphology, lamellar, hexagonal, or cubic. So here again, lamellar, hexagonal, cubic structure. So from the position of the peaks, you get the lattice type, and from the intensity of this peaks, you can get the electron density. This is an electron density structure picture of hexagonal. So you see in the interior, you have high electron density. There, the water is located, and outside you have this hydrocarbon chains, which have a low electron density. So if you insert like some track, you would like increase the electron density in the core, you would immediately see it if you're comparing it with the electron density without any track. So you can locate your track in your nanostructure. Good. Let's just check the time. I just want to make now a short excursion to pressure in the biosphere because when I was working at the synchrotron, one of my tasks was to develop a high pressure cell for biomaterials. And this is a little bit difficult task. And on the other hand, you might ask, why should I study biological samples under high pressure? But if you look into the biosphere, you have also biological systems at 1,000 bar, which is 10,000 kilometers below sea level. And there they also found bacteria which have a functioning bilayer or cell membrane, which can withstand a pressure of 1,000 bar. But this is the deepest point which is available on Earth. So this is near the Philippines. So if you compare this 3,000 meter, I think 3,700 meters is Titanic. This ship sank or imploded, which were diving to find the Titanic wreck, which I think was 2 years ago. So it could only dive to 3,000 or 4,000 meters. But so you have to imagine there is living material existing at 1,000 bar at 10,000 meters below sea level. So for this purpose, we have constructed a high pressure cell, Xray. So I can't want to go into the details, this is technical details, and we have done many successful experiments with this high pressure cell. We have done pressure jumps. We have mapped out phase diagrams. So phase diagrams is a function of temperature and pressure. We have done pressure scans. So we pressurize and depressurize, and we get different diffraction patterns. We have done it with supercritical CO2. So we have dissolved the detergent molecules or amphiphilic molecules in supercritical CO2 and see the changes with high pressure. And we have also investigated LDL, low density protein, LDL, low density protein, lipoprotein particles under high pressure. So lipoprotein, LDL are one component in your blood. So they are transporting the fat molecules in your blood. And they are also responsible, or they are also a cause for atherosclerosis. They are also one cause. And so we have looked what happens with the LDL particles in the normal state and in the oxidized state. Oxidized state means it's transporting oxidized fat molecules. And what happens if we apply pressure? So we see basically, so this is done at 38 degrees, and this is done also 38 degrees. So we start out at 1 bar or 50 bar, and we go up to 3,000 bar. We see mainly two changes. Here in that range, in the inner part of this heparin curve. This range corresponds to the lipid core. So this change is caused by the lipid core, and the inner part is caused by the protein which surrounds this lipid core. So in nanostructure, in the nanostructure space, what does it mean? We have here 50 bar, and at 3000 bar, you see an ordering of the inner core. So the core gets structured, and you see the more structured it gets, the more pronounced the speech gets. On the other hand, the inner part means this happens with the enveloping protein. This gets denaturated. But the main effect is here the ordering of the inner lipid core. If you make the Fourier transformation into real space, you see also before you just have a diffuse, no structure, and the higher the pressure you get, here's this structured, layered structure in the inner core. You can see it immediately here in the B of R function. So this was high pressure with LDL, but we can also do it with lipid alone or also with protein alone. And additionally, we did also high pressure with neutron scattering. Neutron scattering is a different technique. It's the same principle, but you're using neutrons instead of X-rays. And there's also, we see the changes from 1 bar, so the different dimensions of this ellipsoid with high pressure. So there's slight changes also in the enveloping protein structure. Okay, we can also do time-resolved measurements in the laboratory. In that case, we make a slow heating scan. And we want to investigate what happens if we have a phase transition from the lamellar fluid into an inverse hexagonal nanostructure. So in that case, we do maybe 1 minute exposures over several hours, and we heat our sample up. And this is such a time-resolved temperatures in where we go through different phases. And the end, or the conclusion of this, we can map out from the phase diagram temperature versus composition or versus pressure. In that case, we have a temperature pressure phase diagram with a lamellar phase. In that region, we have a liquid crystalline phase, and then we have a hexagonal phase. So slow scans, timers of hand of a heating hands for mapping out phase diagrams. Well, I think I have to speed up a little bit. You can also do jumps instead of small pressure or temperatures, and so you can apply a laser jump. In that laser jump, you heat up your sample within milliseconds, about 10 degrees, so you can make a phase transition within, let's say, 5 milliseconds. You can infuse a phase transition. This is here some examples. So lamellar phase, hexagonal phase. This is cycling hexagonal phase, lamellar phase. So you have to make, this was kind of a synchrotron with millisecond time resolution. And then you can make also you can sketch how this transition is occurring. Like from the lamellar phase, it has to undergo the transition into a smaller lamellar phase, and only then you can proceed into the hexagonal phase. So there is no disorder in between. So there's some disorder. There is basically a transient state, an intermediate state. The intermediate state from here to here is a thinner lamellar phase. So in order to get into the hexagonal phase. Okay, here are some diagrams. So I will skip this. You can also combine small angle headering. When you make a heating scan, you combine it with calorimetry. So we have built, or we had a company which built us a calorimeter, which we put into the SAXS camera. So here's a SAXS camera. We built in here calorimeter. So we measure at the same time there's the heat signal and the change of the nanostructure. So here's such an example. This is a contour plot here, time. So there are maybe 100 frames. Here's angle 0. So here's this headering pattern in the contour plot. So you see the changes, the nanostructural changes. And here you see the thermal signal. So we have here transition, here, and here. So same time, you measure heat signal and nanostructural changes. Okay, here it's the same for another lipid, the type which increase the temperature from 20 degree to 45 degree. This is a lipid called TPPC, 25 to 50. You see the changes here, and here you see the thermal signal. This is a so-called pre-transition, and here have the main transition. You can also measure this in the wide angle reach, but more information you get from the small angle pattern. Okay, here is again some animation. This is what I showed you before, TPPC. You see the change in the spectra. This is small angle, this is a wide angle, and the green line is a thermal signal. See? Changing, thermal signal is changing. Again, another transition. And here you have it for the silver bayonet powder, for this compound. You can measure it also with a two-dimensional detector. So this is very important. So thermal signal and nanostructural at the same time. Another application field is, after COVID, there was a race to find this vaccine, this RNA vaccine. And in order to make this messenger RNA vaccine, you have to pack your RNA. You have to encapsulate it into some lipid nanoparticles. So, but these lipid nanoparticles also have to be stable. And so the research was targeting to find a very suitable composition, which can also be, because if you remember the first vaccines from Pfizer had to be stored at minus 80 degrees. Because above at the room temperature it was degrading, and then Moderna had one which had used at room temperature already. So it's very important also to study this lipid composition under which conditions you get a stable envelope for your messenger RNA. And for that is SAXS a very suitable method, because it just measures exactly that range, the bilayer thickness, or even the whole diameter of this vesicle is in the range of this method. So there's another application. Just an example. So you mix your lipids with the RNA, with the fluidic mixing device, makes this lipid nanoparticles, which has this payload of this vaccine, and then it can study the quality of your vaccine. I think I'm coming closely to an end. Here's just summarizing what you can do by time resolved SAXS measurements. You can do in situ reactions in situ. So you can make either a batch reaction and pump your solution continuously through your Xray beam, and then you can measure nucleation and growth. You can make heating hands to map out phase diagrams, and you can also make a self-assembly. You can monitor by time resolved Xray diffraction or smalling Xray scattering. So there are many possibilities: static and time resolved, static and dynamic. So at the end, I just have brought some, if you want to go further into this technique, there are some books and booklets which are useful for these techniques, and also some web resources if you're interested. There's conferences on this technique. So the last conference on the smalling Xray scattering was just last November in Taiwan, in Taipei. And the next one will be in 27 in Lund, Sweden. So this will be in 3 years or 2 years. Okay, so I have finished my talk, I think, in time. Thank you for the attention, and if you have any questions, you can address it to me.
Thank you, Doctor, for the informative session. Is there any question in the group or anything?
Yeah.
Thank you for your sharing. I believe this one is quite new for most of us in this room.
Yeah. May I ask you to come here because I can't hear from there.
So my question is, because I'm sorry if this question is really simple for you, because this is quite new for most of us.
Yeah.
My question is, previously you said that we might need data maybe from the Xray crystallography data for the structure reconstruction. What if, for example, maybe a new protein in future, then SAXS might be one of the best method because we don't need to crystallize it. Can that be done without the data from Xray crystallography?
Yeah, sure. But I mean, what I showed you, this was only to compare, mostly to compare, but of course, there are many proteins which cannot be crystallized, like membrane proteins. They are very difficult to crystallize, and there you can make but you will never get the atomic resolution like with crystallography. You will mainly get the shape, the envelope shape. You can say it's an elongated, or what we have done, which we compared. The crystal structure was a J form, like the letter J, and we got like an S-shaped form. So we get just the shape, but we don't get the atomic resolution.
Okay. All right.
And of course, if you hand crystallize, you will of course crystallize again and make the crystallography picture. And but the reason why it will go in solution is you can make it time resolved, so you can add some substrate and see the changes in situ. And also for cells which you cannot crystallize, which are like membrane proteins.
Okay. Thank you, Doctor. And just one more question. Previously you said usually we need to actually predict the rigid structure or the solid structure. The static structure. This is more of the protein. then we proceed with the simulation. So, in your case, do you ever encounter, for example, this the same protein? Maybe it was predicted by X-ray crystallography and also predicted by SAXS. And when we subject to simulation, actually it shows a different simulation or different interaction of protein predicted by SAXS and also predicted by X-ray crystallography. Is that possible? Have you ever counted that before?
Yeah, but if when you have the crystal structure, I mean, what was known? What was known? Known?
Yeah. Both are known. Actually, it's predicted by both X-ray crystallography and also predicted by SAXS. And when we did the simulation, will it be the same, or you actually accounted a different interaction between these proteins?
Yeah, I somehow couldn't really understand the question, but what we have done, we were only comparing it with the crystal structure. This was our main purpose. I mean, but in that case, it's already known the structure. There, you don't need the simulation.
I see. Yeah, because for some of us, maybe we need to actually simulate the interaction between the two proteins. For example, when it comes to COVID-19, we want to simulate between spike and also ACE2, the receptor.
Yeah, I mean, what you can maybe verify or falsify, like if you have an alpha helix or a beta sheet structure in your molecule, this you can maybe also find in SAXS. But as I said, you don't have any atomic resolution with because you have an average, because your molecule averages in the solution. So, and you would get maybe atomic structure if you could measure your scattering curve to infinity. But this you cannot do. This is impossible. So you have limited, reciprocal picture is limited for your Fourier transformation. So if you work with simulations, you should rather than do it with crystallography and also NMR. You can also make quite good resolution with NMR, at least with smaller molecules, proteins, and yeah. But anyway, the big advantage or one where you should use SAXS is SAXS cannot do absolute structure. There are other methods better, but what you can see or easily do is to see changes. So this is the best. You apply some external force, either add some substrate or increase temperature or pressure, and then you can see changes. Those subtle changes you can see easily, and very quickly, also very easily. The orbis, and you should also always use complementary methods because you don't get the absolute structure. So you should use electron cryo-EM or electron microscopy to have a complementary method to verify your structure. And it's also more if you have a nanoparticle and you want to solve the structure, you should more work with the exclusion principle. So your model might fit like a cylinder, so it might fit. So you just having a simulation, it fits a cylinder within the limited space. Then you should just say, okay, my experiment, I can make the conclusion it might be a cylinder, but you cannot exclude also other models, slightly different models. But what you can do for sure is, if somebody tells you this model is a disk, and then your SAXS curve does not fit to this, then you can exclude it for 100%. But when it fits, it's a possible model. But not the real one, 100% real one. So use the other way, exclusion principle.
Right. Yeah.
Thank you, Doctor, for the answers. Is there any other questions?
Hi, Dr. Manfred. Cecilia, I'm currently working on liquid crystal, so I just have questions to ask. So after we run the data for small angle scattering,
Yeah.
so is there any tool to assign the peaks to the data, the raw data? Could you can give me suggestion? What are the tools we can use for the peak assignment for the data, for the raw data?
Yeah. Okay. This is more a task for powder diffraction. There are such program, this rich field refinement which you can do. I mean, in our case, like with lipids, the choice of lattices is anyway restricted, or they're not, because in crystallography you have 230 possible space groups. So in with lipids you have only a few ones. So there are programs which you can apply, but there you should more ask the diffraction people who do also diffraction of any inorganic crystals. But there are some you can have some recipe how you do the beam assignment. And maybe lately I have seen also publications where they use AI as artificial intelligence or machine learning to do this. But I mean, with SAXS, the main purpose is not like the powder diffraction people do, is to find the correct lattice. And you also, the number of beats is also limited in SAXS. Maybe you have, yeah, unless you go widening with scattering, but this is also powder diffraction. And these people are more knowledgeable how to do this.
Okay, thank you for the questions, and thank you, Doctor, for the answers. Okay, before we end our webinar, can we have a token of appreciation? Okay, for online meeting, thank you all for joining. So we end our meeting or webinar today. And we have sent out the attendance list in the comment. Please do fill out before joining the meeting. Thank you.